Welcome to this week's Short Clip Friday podcast. Each Friday, I'll be posting 10 to 15 minute segments from previous podcasts. And the point here is just to bring you some of the highest yield topics from each podcast in order to kind of reinforce those topics. This clip is from the Genetics podcast and covers meiosis as well as recombination within meiosis. Enjoy. All right, so moving on to meiosis now. And so meiosis is very similar to mitosis, except for that it is in gametes, which are the sex cells. So I'm going to go through the steps again like I did for mitosis. However, after that, I'm going to harp on kind of these main differences between mitosis and meiosis because I think those are really important to have a really firm grasp on. With meiosis, the acronym to remember is exactly the same, only you say it twice. So it's PMAT, PMAT. So you go through prophase 1, metaphase 1, anaphase 1, telophase 1, and then you go on to prophase 2, metaphase 2, anaphase 2, telophase 2. And so in prophase 1, very similar to mitosis, is the following steps happen. So chromosomes begin to condense and the nuclear envelope begins to break down. Now there are some events that happen that aren't quite the same as in mitosis, and so one of those is synapsis, and so this is where homologous chromosomes line up, and then after this occurs, crossing over of these chromosomes occur, in which they exchange bits of genetic information in a mostly random manner. This results in the creation of recombinant chromosomes. And again, we're talking about prophase 1 of meiosis here. And then in metaphase 2, the spindle fibers attach to chromosomes, and these chromosomes are pulled or pushed into to line up at, again, the metaphase plate. This time, though, homologous pairs of chromosomes line up at the metaphase plate, not just the individual chromosomes. And so if you go look at a picture of this, you'll see exactly what I'm talking about. But again, this time it's the actual homologs themselves, not just the sister chromatids that are lined up. And then in anaphase 1, the homologous chromosomes are pulled apart and start to move towards opposite ends of the cells, and the sister chromatids of each chromosome remain attached to each other at the kinetochore. And that's in contrast to mitosis. And then finally, in telophase 1, these chromosomes are at the opposite poles from each other. And then, you have some of these things that happened in prophase are essentially redone in telophase. So the nuclear membrane reforms, chromosomes decondense, and you continue on and go into meiosis II. And also important to note that in telophase 1, you have cytokinesis. So again, these cells split apart and you form two daughter cells, and these daughter cells are not identical to each other. They are now haploid, so now they have only a single copy of each chromosome. So now moving on to meiosis II. So meiosis II is basically, it is for all intents and purposes the exact same as mitosis. So remember again, you got prophase, which is where the chromosomes condense and the nuclear membrane starts to dissolve. Next, you have metaphase 2, which is where the sister chromatids line up along the metaphase plate. And this time during anaphase 2, which is the next step, the sister chromatids are separated and they move to opposite ends of the cells. And then during telophase 2, you reform the nuclear membrane, the chromosomes decondense, and you have cytokinesis again, where you split into two daughter cells. And it's also important to note, though, that unlike mitosis, these daughter cells are not going to be the same because you had crossing over events occur all the way back in prophase 1. And so these sister chromatids are not exactly the same. They have some different recombinations. And so when they are separated, there's going to be a little bit different DNA in one cell versus the other when they split. So to recap meiosis, so you start with one cell. You go through meiosis 1. You have two different cells that are haploid, and that have these recombined chromosomes, and then those two cells go through meiosis 2, and they split into two more cells. So in the end, you create 4 haploid cells, each with a different DNA, different genes. And so real quickly, I just want to kind of summarize these main differences between mitosis and meiosis. So if we take mitosis as kind of the base case, what are the differences, or what are these different things or processes that occur within meiosis? The first is synapsis, and so this is where the homologous pairs of chromosomes kind of meet up, and then once they're met up, they exchange bits of information, bits of DNA, which is called crossing over. And lastly, the cells that are produced by meiosis are haploid, which means that they only have a single copy of each chromosome. And the last thing I want to mention is how meiosis produces lots of genetic diversity and how these gametes are all different from one another. So this genetic diversity comes from two main points in meiosis. So when crossing over occurs, it's very random. So you might have a little bit of DNA from this portion of the chromosome that moves over to this portion, and then you have a different portion that's swapped in this chromosome. So different chromosomes cross over at different points. It's random. So that's the first factor that contributes to genetic diversity during meiosis. And then the second is that these homologous pairs line up or orient in a random way. So in metaphase 1, as I said, these homologous chromosomes line up at the metaphase plate. And so you might have one of these line up on the right side of the plate, the other the left, or you might have the other way around. And then when these chromosomes are pulled to the poles and cytokinesis occurs and these individual cells form, the chromosome that was on the right is going to be in the cell on the right, as the chromosome on the left of the metaphase plate is going to be in the different cell. So that lining up is random, and so you're going to randomly receive one of these chromosomes. And there's 23 different chromosomes, so you're going to receive kind of a random assortment of those 23. So some are going to go to the cell on the right, some to the left, and again, that lining up is random, and that contributes to genetic diversity. And just to illustrate this point, this random orientation of homologous pairs of chromosomes alone, without even considering crossing over, allows for over 8 million different types of possible gametes. So just keep in mind that alone contributes a lot to genetic diversity.